From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 53, recorded on July 1st, 2024. I'm Vincent Racaniello, and you're listening to the podcast about the nervous system. Joining me today from Salt Lake City, Jason Shepard.
Hi, Vincent. Welcome back.
Thank you very much. Good to be back. Also joining us from NYU in New York, Tim Chung.
Hi. Yeah, welcome back, Vincent. It's been a while. It's been 2 months.
2 months, you're counting, huh? Well, yeah. I'm here. And from New Orleans, Vivian Morrison.
Hey, everybody.
Good to be back with everyone. We have guests today from across the Atlantic Ocean from the Champalimaud Foundation. Joseph Paton, welcome to TWiN, Joseph.
Thanks for having us.
And Felipe Rodrigues, welcome.
Thank you.
If you enjoy our podcasts, including TWiN and all the others, we'd love to have your financial support. Microbe TV is a nonprofit entity, which means in the U.S., your contributions are U.S. federal tax deductibles. So please go to microbe.tv/contribute. So let me understand the relationships of you gentlemen. Joe, you're the PI, is that correct?
Yep, that's correct. And
Philippe is your PhD student?
He was, but he recently defended his PhD, so now he's a postdoc in the lab.
Moving up in the world, right?
Debatable.
Yeah, but it still feels like
moving up in the world. As the one closest to postdoctum other than Philippe.
Debatable. That's very good. Let's start out. We're going to talk about your paper today, all about temperature and timing. But first, let's get a little bit of your histories. Joseph, I don't think you're from Portugal, are you?
I am not, but I've lived here for, now 16 years. I came here right after finishing my PhD in New York, where Tim is, at Columbia. I did my PhD in neurobiology and behavior, and then was sort of casting around for sort of independent postdoc positions, various places in the U.S., and one of the places was Cold Spring Harbor, and Zach Mainin, who was there at the time, was being recruited to start a new neuroscience program here in Lisbon at this recently formed foundation called the Champalimaud Foundation. So I came here 16 years ago, thought I would be here for 5 years. 16 years later, I'm still here.
I guess you like it, right?
Yeah, exactly. It's just that as the place grew and as we recruited more people, I realized that there weren't really places in the world that were better for the kind of science we were doing. So here I am.
And in fact, the center, if I'm not mistaken, it is actually called Champalimaud Center for the Unknown.
That's correct.
Is that right?
Yeah.
Oh my
So
gosh.
it tells you about the ambition
of
the center. Yeah, it's amazing.
I like that a lot.
The unknown in neuroscience, or the unknown just in general? Like, does it have other?
The unknown
domains?
in general. That's hearkening back to Portugal's history of explorers,
right?
And but applied to the modern age in science.
Joe, what do you have around your neck there?
Oh, sorry. This is because we're in the midst of the Euros right now, and later today Portugal's going to be playing Slovenia in the round of 16. So I'm just getting prepared.
We have to make sure our sense of timing is on point today. That's what we're
correct.
Will be good.
Yeah, Jason, you got your work cut out for you.
Philippe, what's your history?
My history is shorter. I'm actually from Lisbon. I grew up here, and then I studied biomedical engineering. At the end of it, I wasn't really sure whether to go into industry or more research. But then I just sort of applied to a number of labs at the Champalimaud, and was lucky enough to be offered the chance of going there as a technician in Joe's lab. And so that's kind of how I started. And then I liked it so much that I stuck around for a PhD and now a postdoc and life, I guess. Let's see.
Here I was thinking we were special, Philippe, but turns out you just applied, you just spammed everyone.
Right place at the right time.
So the Champalimaud has a PhD program, or is it affiliated with an academic institution?
Yeah, so we run our own now several PhD programs. The PhD program in neuroscience was the first program. We have partnerships with degree granting institutions, namely the New University of Lisbon and the Institute of Applied Psychology in Lisbon. So we run our own PhD program, sort of like a hybrid American European style PhD program. We craft the curriculum, but then the degree is actually from one of these universities.
Mm-hmm. So where's the PhD in the unknown?
Yeah, well, that's once you figured it out and you get your PhD.
I see. Then it has
no
a name.
longer
Okay.
unknown.
Is it a time limited PhD like it is elsewhere in Europe and around the world, or is it like in the United States where you don't know how long you're going to be there?
It's a constant battle. We aim for 5.5 years on the PhD. We have an official limit of 6 years, but we try and let the science guide the timeline, and abide by best practices in terms of oversight. So students don't fossilize here.
6 years is good. I mean, I think that's the aim of most PhD programs here too. But we have to do the rotations, which takes a year. So in effect, they're only actually in the lab like 5 years. But
yeah, same here. That's what I meant by hybrid, because sort of the traditional system in Europe is you apply directly to a lab. You don't have rotations. Right. You don't have an initial period of coursework. So we have a sort of accelerated version where we have coursework and rotations for the first 9 months, 10 months or so, and then you start your PhD. Right. In the lab.
So any students listening, if you'd like to apply, here's the link.
It's an awesome program. You can ask Philippe if you want the lowdown.
Speaking of timing, what an interesting lead-in, Vivian. Thank you very much.
No problem.
We're going to talk about your paper today, Nature Neuroscience, using temperature to analyze the neural basis of a time-based decision. And I want to start off by first sentence in the paper. Much of behavior is dependent on time. What does that mean?
Philippe, you want to take that?
Sure, I can take a first stab at it. I think it refers to this almost trivial fact that behavior unfolds through time, and so systems that evolve to control that behavior have to have dealt with time in one form or the other, and in some cases leveraged the regularities in the temporal statistics of the environment to sort of benefit fitness in some extent. And so it's really the scaffold together with space maybe of all behavior.
But this is not just kind of, because when people mention time, like we have to pay attention to the regularity of time. Maybe the most obvious one is just we have a 24 daylight cycle that we have, and it's fixed, and our body is very much attuned to that. And in fact, inside every single cell in us has a little circadian clock that can be, kind of tuned to the circadian rhythm. But the kind of timing that we're looking at is more flexible and more kind of maybe equally important to behavior in terms of this kind of timing is, instead of 24 hours, kind of very periodic, it is something that we can learn from the environment based on, for example, how long. Actually, in nature, what do you think the kind of timing that you guys look at? When would it come into play? I
think the kind of technically the kind of time range we study falls under the umbrella of interval timing, which is timing in the range of seconds to minutes. And so I think that's where the most interesting part of timing happens, because it's the one that, like you said, it's not governed by some large celestial body that's going around at a periodic rate, but instead it's about how it can be adaptive to leverage arbitrary relationships in time in this kind of range to adapt essentially to volatile environments, to other agents that are also capable of learning those temporal statistics and benefiting from them, and sort of this interplay at that
this
range of seconds.
This is, I mean, stepping back, this is a fundamental thing that brains do, which is to order events, right? It's not even about how timing is encoded in the brain. It's making sense of the world. And I mean, correct me if I'm wrong, but the interval timing paradigms that neuroscientists use, it's more about that it's something we can measure. It's something we can control, but we're still trying to figure out how the brain orders information so that it can actually learn, right?
Yes, although I think in the spectrum in the spectrum of reduced behavioral tasks that sort of lack ethological parallel, or that have a very clear one, I think interval timing actually, maybe some of the tasks at least, like peak procedure tasks where animals have to sort of infer how often a reward is coming on a fixed interval, for instance. Those, I think, are very easy to map onto foraging behaviors, where there's some regularity with which a particular patch becomes valuable, and so adapting on a relatively fast timescale to that refuel or replenishment rate, I think is very easily mapped onto the kinds of reduced behavioral tasks we do in the lab. But maybe some other ones become more artificial and maybe harder to link directly with some natural behavior.
I wasn't saying that they're artificial. I think it's just more that this is the way we're getting at timing.
Right.
But so it's not just... Sorry, please go ahead.
No, just going back to what Jason was asking. I mean, sometimes in talks I'll use the example of space and vision to kind of illustrate the point you were making, right? If I look at an image, it's made up of, pixels with certain luminance values and, color spectra at each location, right? So there's structure in the kind of spatial arrangement of information. Similarly, in the time domain, there's sort of structure in the occurrence of events or behaviors in relation to events, right? And that structure contains information that animals can use basically to produce more adaptive behavior that's going to benefit them more, right? And so a lot of that has to do with not only just ordering things, but understanding the temporal relations between things. So for instance, learning cause and effect relationships,
right.
Just like if you were to take an image and scramble all the pixels, right, you would no longer recognize what's in that image, right? Similarly, if you took all time points of experience and just randomized them, it would be a complete jumble, right? And you would lose all of the information that we think of as kind of creating our reality, right? So it's really very fundamental. I think that's the thing that Jason was pointing out.
But I think not just because like in terms of structuring our behavior, it's very important to remember what things happen, like whether thing A happened before thing B happened before thing C, so the sequences of it. But in timing, there's actually something extra, which is not just what happened before something else, but how long was it? So a lot of this structure actually, by just observing, when we're sometimes it just appears without us looking for it. For example, if you do the kind of Pavlovian conditioning that on the podcast we sometimes talk about, like you ring a bell and then after 5 seconds you present the mouse with a little bit of juice or a bit of water at the liquor, what you see is that if it is a 5-second delay every single time, the mouse would eventually learn to not lick when the bell comes on, but lick close to 5 seconds. So at the beginning, when the bell comes on, there's never any reward. But as the time goes on towards 5 seconds, the reward chance of the reward appearing is increasing, and the mouse accordingly licks faster and faster. And that tells you that there might be some sort of mechanism inside the brain to tell the mouse what time it is. And it's actually, I remember, so I never actually ever talked about my PhD research because it happened so long ago, I almost forgot. But I actually did my PhD in timing as well. And when I first went to my PhD, when I first entered my PhD, I was very naive, and I thought, you think about the story people say that goldfish only has like 7 seconds worth of memory. But the task that our lab was training was to have a rat. So we used rats back then, and a light would come on, and if the light has been on for less than 25 seconds, the rat would have to go left to get some water, actually get some food pellets. But if the light has been on for longer than 25 seconds, the rat will have to go right to get the food pellets. And the rats were able to do it, able to learn it. And that was absolutely mind-blowing to me, that the rat, something like rat, that we didn't even tell it what the rule it was. It didn't know what 25 seconds mean, but it can learn some of the behavior like that, which is absolutely incredible. And that actually is very similar to the task that Philippe and Joe looked at in the paper we're discussing.
So I want to ask, I have a rudimentary understanding of timing in biological systems. Like I know cell cycles are timed in mitotic cells, right, by cyclins, by levels of cyclin-dependent kinases and so forth. In virus-infected cells, the timing of the infectious cycle is manipulated by the levels of, in some cases, transcription proteins. So do we know in the brain what kinds of mechanisms are involved? Is there a clock, like a processor of a computer clock?
So that's kind of what this paper was about, or this basically the original set of papers that then triggered this one. This whole body of work has been aimed at, in a way, answering that question. I think there's very good evidence to say that the duration or the time between events is something that is not necessarily explicitly encoded by a single neuron or some cellular process, but rather distributed in the dynamics of populations of neurons. So one analogy we've used in the past is to think of sort of a pond of neural activity, where if you drop a pebble in that pond, it's going to ripple out with these dynamics on the surface of the water that eventually die out. And if you were asked how much time has passed since the rock hit the surface of the water, just by looking at the ripples, you'd do a fairly good job at saying, was it more than 1 second, less, something like that. And so you could think of an analog of that in the high-dimensional space of population dynamics in multiple areas across the brain, where some stimulus triggers a sequence a sequence of activity states from which you can decode how much time has passed since event A or something like that. Yeah.
I think we have to thank Dean Buonamano, or credit Dean Buonamano for that for that analogy of the stone in the pond and the ripples. But yeah, and the idea is not, again, like Philippe is saying, the idea is not that this is like a clock where the central function of these dynamics is to encode time. It's just that almost all computations that the brain has to perform kind of rely on having a temporal basis, right? if you have to detect structure in the world that's temporal in origin, then you need some way to kind of tag individual events or actions with, sort of a timestamp. Or if you need to produce some time varying function, like in the context of motor control, then you need some basis with which to kind of construct those time varying functions, right? So it's just sort of, we think of it very much as being implicit, this kind of timing. Of course, I should say the caveat is that there are multiple forms of timing that play out in the brain, right? You already mentioned circadian timing. The suprachiasmatic nucleus has these kind of negative feedback loops between transcription factors, like you have many cells in the body that then get entrained by light, right? So there are multiple kinds of temporal processes that occur in the brain, and what we're really focusing on is these sort of temporal processing that's mediated through population dynamics.
Well, I'll put a plug in from my colleague here at Utah, Jim Hayes. They just published a paper, also in Nature Neuroscience, suggesting that there are cells that could be clock-like, in that their firing activity is sort of fundamental to the circuits that are doing this for the interval timing. And so I wouldn't say that we've ruled that out, that there isn't cells in the brain that act like clocks in some way, partly just due to their firing pattern. Right.
Love Jim. Say hi to Jim. But I think, even Jim would say he thinks about, that kind of temporal process in the context of what he thinks, medial entorhinal cortex is doing as a whole, right? I don't think he would push the idea that this is a master clock, that is imposing its time base on every other process in the brain. So that's the way we like to think about the timing signals that we record in the basal ganglia, is in relation to what we think those circuits are doing.
I think it's a bit of a chicken egg, and this is the problem, right? What's the causal? Where is the intrinsic timing coming from? And that's the big question, probably.
So let's dive into your paper. You use temperature to look at timing in this paper. Maybe you could explain to us how that works.
Sure. Should I go for it?
Yeah, you take that, Philippe.
So basically, there was this this paper that preceded this one, where this eLife paper, where Tiago Monteiro and Thiago Veia saw that the speed of striatal dynamics during this task, this interval categorization task, was correlated with the timing judgments that animals made. So if dynamics in the striatum were faster, animals reported long more often. If they were slower, they reported short more often. And so we wanted a way to see if this was really, this relationship was causal in nature, so that if speed was actually driving variability in timing judgments, we needed a way of causally manipulating this feature of the activity, so the speed of these dynamics, without disturbing the pattern. And so if you think about it, this is not something that's trivial, even with the amazing methods we have access to, like optogenetics, where you don't want to just amp everything up or inhibit everything. You really just want to play with the speed while preserving the pattern of activity. And
just a very brief note, maybe it's worth explaining to the audience what we mean by neurodynamics. And I'll take a very quick crack. Please correct me if I'm wrong. But basically, as the mouse is, so you put a mouse in or a rat, an animal in a timing task. So the rat's job is to basically time how long a stimulus duration has been, and then go to the left versus right if a duration is short versus long. And what you see inside the brain is as the trial start, as the duration starts, there's a signal to tell the mouse to start timing. You see kind of a pattern of firing that is somewhat, I wouldn't say completely stereotyped, but has some structure that is kind of repeatable with some noise, with some variations. And it kind of, like you said, with kind of ripple in the water, it unfolds in time, kind of with some regularity from trial to trial. And this pattern of regularity of firing of all these cells together gives you a kind of global pattern. And this is the dynamics, how the kind of the firing rate changes from cell 1 to cell 2 to cell 3. That's kind of the dynamics you're talking about. And by speeding it up versus slowing it down, you're talking about how, cell 1 to cell 2, not quite passing the baton, but, how the whole shape changes. You can squish it or stretch it. That's the goal.
Yeah, what a useful simplification is to think of just a sequence of activity where neuron 1 fires, then 2, 3, 4, and so on, even though it doesn't have to be strictly a sequence. Because like you said, the key thing is that these dynamics as a whole, their pattern on average is reproducible. So it happens every time you provide the stimulus, and it's unambiguous. So in the range of time that the animal needs to use information about time to go left or right, the pattern cannot repeat itself, because otherwise it becomes ambiguous whether a given state should be followed by moving to the left or to the right. So
which is different from the clock, because the clock's tick will be the same every single time.
Well, except that if you think about the hands on the face of the clock, right? The state of the hands doesn't repeat itself for a 12-hour period, right?
Yep.
And then if you have the AM PM, right, then it's for 24 hours, right? So the same is true of the state of a clock face as, has to be true of these neural dynamics.
We're going back to even simpler. So all the temperature is doing is slowing down or speeding up the physiology of the cells, in that the firing. So the idea is that the firing pattern is the same, but it just slows or speeds up the sort of firing pattern. But the actual pattern itself is the same.
Not exactly, and this ends up being very important. So I'm glad you raised this point.
But it's exactly how we thought it would work going in.
Right. That's what we were going for. And it's actually kind of astounding if you step back, because, every protein, every, lipid, every element of the biological system that makes up the brain has some temperature dependence, right? And there's nothing that guarantees that changing the temperature, given all those diverse dependencies, would actually lead to, slowing and speeding of population dynamics, right?
But on a kind of like a molecular, on a biology, maybe chemistry level, you would expect things to slow down. That's why we use ice buckets.
You might accept that. For example, if you have a certain, set of constituent ion channels, right, in a cell, then and they have their own particular temperature dependencies, then you can have kind of unexpected impacts on, for instance, excitability, which might, change the time course of patterns of activity. Right. So it's important to check and see what you're doing with temperature, which is something that Felipe and the other authors did.
Right. I mean, and electrophysiologists have known some of this for a long time, because, of course, you take slices from an animal and you record from them, and there's actually a lot of discrepancy in the field, partly because people will record at 32 versus 37 degrees in their slice chamber, and that can actually affect the kinetics of a lot of these electrophysiological properties. And so it's cool that you're looking at this in vivo and that it actually worked.
So how do you want to use temperature to manipulate the timing? How do you do that practically? What was your experimental design?
So this was an iterative process. There were a lot of versions to the final device that we ended up using. And all of this was started by Tiago Monteiro, which is another first author in this paper. He started the whole project, built the first prototypes with tiny fans to sort of help with these heat dissipation and all sorts of crazy things. And then Margarida Pschirra, who is another first author in the paper, eventually developed or polished the final version of the device that we ended up using. And
our hardware software platform. Got to give a plug to Champalimaud Research hardware software platform. They're incredible.
Yeah. But without going into too much detail, we use this Peltier device that leverages the Peltier effect, which allows you to produce temperature changes by applying current to this device in one direction or the other. And with this, you're able to get bidirectional manipulation of temperature at this small device that you can hook up to a probe, in our case a silver probe, that then targets your target brain region, which in our case was the striatum, so the major input of the basal ganglia, and also as a control the primary motor cortex in another set of experiments.
Sorry, I was just going to say
no.
There's a history here, of course. So like I think the first paper to use this kind of method was back in like 2008 or 2009 by Mike Long in
here at NYU. Well, he wasn't at NYU then, but
now he's at NYU. He was at MIT. Yeah.
Well, I mean, if you look at the title of our paper, it's actually an homage to Mike's work, who's a friend. Philippe, do you remember exactly what the name is? It's using temperature.
It's using temperature to analyze temporal dynamics in the songbird motor pathway.
There you go. Right. So we tried to use a similar form as a means of paying homage to that kind of seminal work. But the use of temperature, it goes even further back. I mean, largely it's been used as a way of really kind of temporarily lesioning or shutting down certain areas.
Right.
If you cool a lot, then you can really kind of shut off neural circuits.
Right.
But this more graded, smaller magnitude temperature manipulations, is more recent.
I didn't catch it. Yeah, that is pretty cool that the title is almost exactly like that.
It's like Alfred Hitchcock, right? It appears in his own movies. Okay.
Oh.
So you implant this Peltier device. These are rats, right? You put it in.
Yes.
Another thing you did, which I would like you to explain because I don't understand it. You deliver channel rhodopsin with a viral vector. Why do you do that? What's the purpose?
So this was in there are two main sets of experiments in this paper. One are the ones where we manipulated temperature in the striatum while animals were performing our interval categorization task, so during behavior. And the other was in anesthetized animals, where we manipulated temperature in the striatum while recording neural activity there to verify that what temperature was doing was slowing down dynamics and speeding them up, depending on whether you were cooling or warming. But a problem is that if you're recording from the brain of an anesthetized animal, there's not really anything to align neural activity to. There's not in behavior. There's always the onset of the stimulus, and that's where you align your activity to then see if those dynamics are scaling or not. But in this case, we didn't have that, and so we expressed channel rhodopsin in the ventrobasal complex of the thalamus, which provides both direct and indirect input to the dorsal striatum to both provide reliable dynamics and give us an obvious point on which to align neural activity and then assess what temperature was doing. So and this was something that was adopted. Sorry, just very quickly. This was a protocol that we adapted from the Juedo-Orozco lab that they developed for a different set of questions.
But it's go back to your analogy. This is basically by optogenetically stimulating the thalamus, you are throwing the stone in the water so that you can then see the ripple. Yeah,
exactly. Yes.
Yeah, we can get into
both see the ripple.
Sorry, go on, Philippe.
I was just saying both see the ripple and then see what temperature does to that ripple.
Right. Yeah, that's what I was going to say.
But yeah, I'm actually. It is actually very incredible. So it's it's impossible to do it on a podcast. But if you look at Figure 1, it is so striking that you really, as you lower the temperature, so as you throw the stone in, or as you kind of optogenetically stimulate the thalamus or ping it, you see like neuron firing at the beginning, very, very kind of time locked to the stimulation. But then after a gap of maybe like 150 millisecond, they start kind of almost reverberating. I don't know whether that's a good word to use, but they kind of, there's a second volley of firing and the neuron kind of start pinging away. And then as you cool it down, this whole thing stretches out. So it takes longer for the same pattern to kind of arrive. The whole thing stretches out. And as you heat it up, the whole thing is like squished together. And it's really incredible that you got such a nice, repeatable behavior. Not behavior, sorry, that neurodynamics. Yeah, it's
and this the cool thing about the paradigm that Pavel Ruedo-Orozco developed is it, in contrast to dynamics that you might record during behavior, which are sort of full of all the variabilities inherent in behavior, this allowed us a paradigm with which to produce extremely reliable dynamics over a relatively long period of time of up to a second or so.
Yeah.
So we could really then carefully examine the impact of temperature on those patterns of population activity.
Mm. So if you these initial experiments with the channel, how could you summarize what you found there?
So the main part of that summary is that we could actually use temperature for what we wanted to do then in the behavioral experiments, because it really seemed like temperature was slowing down dynamics when you lowered it and speeding them up when you increased it past the control sort of basal level. But then there was this second effect of temperature that we had not necessarily anticipated, and which is the thing Joe was foreshadowing before, which was that basically any deviation from control, any deviation from 36, 37 degrees, seemed to lower baseline levels of activity in general. So across the populations we recorded, firing rates just went down, both when we warmed and when we cooled. And this was initially felt surprising. I guess you always expect monotonic effects, but a bit fortuitous also, because if both effects happen to be monotonic, then we would be in a tough spot to attribute the effect of our manipulation to one or the other. And this actually made us more confident that any monotonic effects that we might see on behavior was driven by the monotonic effect of temperature on the speed of neural dynamics. And it actually made us go and look for non-monotonic effects of temperature, which we may then attribute to this other effect on this sort of lower dimensional baseline level of neural activity. Yeah.
Okay, why don't you describe the behavioral experiments for us now?
So it's very similar to the kinds of tasks Tim was describing in the beginning. In our case, it's the stimulus is auditory instead of visual, and we used empty intervals instead of filled intervals, meaning that instead of a light being on and then the animal has to go one way or the other depending on how long it was, we do a sequence of 2 beeps, so beep, beep. And then if the silence between the 2 tones was more than 1.5 seconds, they need to go to the left, let's say. If it was less than 1.5 seconds, they need to go to the right. And they become really, really good at when the stimulus are easy, they never make mistakes, essentially. They always say that a very short stimulus is short, and they always say that a very long stimulus in relation to 1.5 seconds is long. But then as the stimuli get closer and closer to the category boundary, behavior becomes more and more variable. And that's where we had previously seen, so in those stimuli where sometimes everything's the same, but sometimes they report short, sometimes they report long, that's where we had previously seen that when they report long, it seems that activity was going faster in general, and then when they report short, it was going slower in general. And now the goal was, okay, let's manipulate temperature and then see whether we can induce these biases, regardless of endogenous variability in speed.
And that kind of makes sense, because if you think about it, if your internal neurodynamics is running faster than usual, so it's kind of like you're kind of I don't know how to describe it. You're kind of like Superman. You see things very, very. The whole world around you seems to move slower relative
So
to yourself.
you have an attention deficit disorder, you mean?
Perhaps.
Because that's our experience, is that everybody else is moving much more slowly.
Oh, we need to
Yeah,
we
so
need to see
we
whether
need-
ADHD humans have like temporal deficits in these sort
Oh,
of tasks. I
absolutely.
don't know if Joe, you know.
I don't know about in these tasks. That was one of the
Okay.
things that this paper made me think of as I was reflecting on my own experience and of time blindness and time elasticity, and just consistently misjudging how much time has passed or being able to estimate how much time a task is going to take. But I do think that is quite different than the tasks
And
that they're
do
testing
you
here.
notice?
So
Maybe dovetails to some of other research that Joe does in his lab. Do you notice if Adderall or like dopaminergic drugs would
there's
alter
a
things like this?
2016 science paper that shows that they show
Yeah.
that by modulating, stimulating, or inhibiting dopaminergic neurons, you can influence behavioral sensitivity to time, and that. So I have that open on my computer to read.
Yeah, and in fact, I mean, lots of different disorders that are thought to involve the dopaminergic system also affect various different aspects of timing behaviors, including ADHD, but also Parkinson's, for example. And there's a lot of kind of interesting work still to be done that we have lots of thoughts on and ongoing experiments about, related to kind of how dopaminergic modulation of striatal circuits affects various different kinds of temporal processing.
But I cut you off, Tim. I think you were.
Sorry, I was just kind of developing what Philippe was talking about. So basically, if your internal neurodynamics like is running faster, you would judge the rest of the world around you for being slower. So any duration that used to be 1.5 seconds, you would think, my God, I've been waiting here for 3 whole seconds. Can you get a move on? So you would tend to respond to, like, you would tend to go to the long, the choice that is designated long. Whereas if your internal dynamics is going slower, the 1.5 seconds would elapse, and you're like, whoa, did that just happen? It seems like no time has passed. So we go to the short side. And Philippe, correct me if I'm wrong, you guys previously found in your eLife paper that if a mouse is mistakenly choosing the long side, the internal neurodynamics seems to be running faster. And conversely, if the rat, I might keep making that mistake. If the rat is mistakenly choosing the short side, it is correlated with the neurodynamics running a bit slow.
Yeah, exactly. I wasn't on that paper, but that's exactly it.
Okay.
Exactly. Though I might just say that this, I think the thing you were saying about how it might feel to have your dynamics run faster or slower in one particular brain region, I think we tend to stay agnostic to that, because it kind of gets into perception, which we then tend to see through the human lens, of course. But this may be something that's actually very brain area specific and very task specific. So you could imagine that endogenous dynamics in the striatum in the context of these tasks are running faster or slower on a particular trial, and that determines that the animal chooses long or short with higher or lower probability. But then those do not have to necessarily be coupled to dynamics elsewhere that may also be varying in the speed with which they're evolving. And so if things are uncoupled, then it becomes trickier to say something about the global experience that the animal or the human is having. In this case, we tend to think of these dynamics as representing something that could be used to inform what action to take and when. So it could be very policy oriented and not so much perception or experience related. I don't know if that was clear.
Yeah, point taken. Sorry, I was trying to picture how to explain to you, but you're absolutely right. It can be completely, and of course, the basal ganglia is very motor related, so it might be a very motor dependent timing kind of thing.
But it's a useful analogy.
So tell us what you found as to the effect of temperature on movement and temporal judgments and so forth.
So in a nutshell, if you cool the striatum, they report short more often for any given stimulus. And it was really striking because for all stimuli, all animals, we saw this relationship, this monotonic relationship where if you cool, they report short more often. It's not deterministic. It's not as if you cool, they always say short.
It's not all stimuli, right? It's more for the more difficult stimuli, right?
Oh yeah, sorry. I was thinking about the psychometric shifts. It's true. It's not for all stimuli because it's not deterministic. So because you don't, if it's not the case that you cool for any stimulus and they always say short. And so for stimuli that are very far away from the boundary, like the very long one, for instance, by cooling, maybe you speed things down and the time basis they're using to guide their decisions is running slower, but because there's so much time after the boundary, they do end up making the correct choice. And so for those that are very far away, you do not see an effect, but the ones where any kind of variability may be consequential, for those you do see this robust monotonic effect where cooling, higher chance of them reporting short, warming, higher chance of them reporting long.
And that's important. That's important, right? Because if it had the same effect on all stimuli, right, that would be kind of maybe consistent with just inducing like some kind of motor bias, right? The fact that you're getting this consistent bias specifically for those stimuli that are closer to the decision boundary, that's what's consistent with you actually having changed the kind of latent representation of that dimension we're asking animals to make a judgment about, which is elapsed time, right? Or latent representation related to that dimension.
Does this have something to do with why you guys had the fixed versus non-fixed or like free exploring components? You had those two different conditions. I didn't understand if you were trying to like dissociate any bias that might occur because of the movement from.
Thank you to reviewer 3.
Oh
wow. No, I mean, so actually this ends up being relevant for things maybe we'll get to, which is that there has been sort of suspicion within the timing field for a long time that, sorry, it's hard to talk about time without saying the word time multiple times, but that the brain might offload a lot of timing function onto the motor system. There's a lot of evolutionary pressure within motor circuits to kind of embed the capacity to create really reliable time-varying patterns of activity, of course, to control behavior, right? And people had been observing for decades that animals will often kind of develop sometimes what are called kind of superstitious behaviors. They'll develop like particular time-varying patterns of activity in timing tasks. And that's led people to speculate that they might actually be kind of externalizing the clock in some sense by, kind of learning to produce some dance, right? And then just keeping track of where in the dance they are in order to estimate some time interval, right?
Like
And
counting
so,
Mississippis.
like counting Mississippis, exactly. And so, we were open to that possibility. In fact, we published a paper in 2014 where we pointed out that there were behavioral correlates of the animals' decisions. Although in that 2015 paper, we showed that the information about the decision showed up in the animal's behavior several hundred milliseconds after it showed up in neural activity. So that at least suggested that, it wasn't on the final path out of the brain, that neural activity that we were using to kind of decode information about the decision, but it still could be used for kind of like motor planning or something like that. And so, we really wanted to see, and particularly reviewer 3 wanted to see that what we were looking at in terms of the population dynamics was not just simply reflecting ongoing patterns of behavior, right? So we implemented these 2 versions of the task. One where the animal was less constrained; they were free to move about during the interval period. And again, oftentimes animals will develop kind of stereotype patterns of movement. We observe the result there in terms of the speed of population dynamics relating to the animal's choices. And then we have another version where the animal is forced to stay what we call fixating with their snout in the initiation port during the entire interval period. Now, of course, they can still be producing some kind of, sub-threshold patterns of behavior that we're not really observing.
Like counting Mississippi onto their breath.
I was going to say.
Right, exactly. So, it doesn't completely prove that it's not a quote unquote embodied strategy. But at least the sort of main features of the result are there in the face of these big differences in the degree to which the animal is free to move around. So
I'm actually
that's
quite
why we have those
surprised.
2 versions.
I'm quite surprised when the version of the task where you allow the animal to run around, do a dance, and therefore kind of time by using its movement, they didn't do better. If anything, maybe the variability is even higher. So it's kind of interesting. Yeah.
Yeah. And in fact, not all animals actually do run around the box. Some do, some don't. And there's not any obvious difference in performance across the 2.
Yeah. It kind of makes sense because you really do. I don't know whether if it's fully embodied, like if you really time by sort of kind of gesturing and movement, then any kind of stimulus, any environmental constraint would completely mess you up in the future. So it actually makes sense to have some independent way of timing inside your brain.
Yeah, and we think of the brain as sort of like a 747. It's got a lot of redundancy and it's opportunistic. And if there are regularities in the environment or in behavior that the brain can latch on to perform particular tasks better, it will. But that's why we do controlled experiments, where we try and kind of remove those regularities. And and I think we think in general of the brain as sort of an open dynamical system, right? It has its own intrinsic dynamics, and then it's subject to, the time varying inputs from the environment and reentrant inputs that are the consequences of its own behavior, right? But so in most cases, the time varying patterns of activity that you see in the brain are a product of those 2 things, right? The inputs the brain is receiving and also its intrinsic dynamics.
So you guys found that, cooling the brain down makes the mouse choose short more often, and then heating the brain up makes them choose long more often, kind of completely matching the neurodynamics inside the dorsal striatum. But then you found that, so in a kind of monotonic fashion, right? So like cooling the temperature makes you go short, heating up the temperature makes you go long. But you guys found, interestingly, slightly more complicated effect on the behavior in terms of the movement speed. So could you tell us a little bit about that?
So I think maybe the first thing to say about how temperature affected the kinematics of animals' choice movements was that it for sure did not scale them up in time in a monotonic way. So that was the most striking thing, was that there was no obvious temporal rescaling of either the paths that animals were taking to reach the choice ports, or how fast they were going to reach those ports. But then there was this more modest effect, so not as clean, perhaps, as what we had seen in terms of the effect of temperature on the discrete decisions animals were doing, which was, like you said, this monotonic effect that nicely matched the monotonic effect of temperature on the speed of striatal dynamics. But there was this additional, more modest effect where it seemed that temperature was affecting just the average speed with which animals were reporting their choices in a non-monotonic way. So any deviation from control, they just got on average a bit slower, which we were tempted to map onto the non-monotonic effect that temperature had on the average activity of striatal neurons, independent of how fast they were going.
And this is
yeah.
Sorry, keep going.
No,
I didn't mean to cut you off.
Go.
I was just going to say, there's other data out there collected in a variety of different contexts that, has shown that these same circuits are involved in what you might call motor vigor. Okay. So, one of the cardinal symptoms of Parkinson's disease, for example, where, the main kind of causal driver of Parkinson's disease, we think, is the loss of dopaminergic innervation of dorsolateral sensory-motor striatum, is bradykinesia, or slowness of movement, right? And this has been shown to be really a problem with the kind of motivation to move vigorously, okay? We also know in animal experiments, if you just inhibit one of the feed-forward pathways in the striatum, the direct pathway, you can produce simply slowed movements, but where the structure of the movements is the same. So we know that this is a structure that's implicated in how vigorously movement is produced, right? And it's just interesting that that is a form of temporal control of behavior as well, right? But it seems to be driven by what, at a population level, looks like a different mechanism, right? And that was one of the really surprising kind of discoveries of this paper. The first order thing that we've been talking about, you rescale the time course of population activity, and you systematically rescale the animal's patterns of judgments. That was the hypothesis we set out to test, right? And then this was a real surprise.
So do you think, so you mentioned earlier at the beginning that one good thing about cooling the brain is that you can affect the neurodynamics. But I'm guessing the reviewers didn't ask you to do this, but if you were to actually reduce the excitability of striatum so that the cells will fire less, do you think you would get the dissociation where you would reduce the firing rate of all the cells without affecting the neurodynamics? And would that give you kind of same timing sensitivity? So no messing up the duration, but still, the rat would respond slower, that kind of stuff.
Yeah. So I think we suspect that might be the case, because after we saw this, we saw that there was this non-monotonic effect on movement speed, we went back to the neural data we had recorded and looked to see if variability in baseline levels of firing correlated with the animal's movements in some way. And we found that there was this non-monotonic, relationship with reaction times, where the lower activity was before the animal initiated the trial across the population. Deviation. The lower activity was, the higher the reaction time was, when then several seconds after, when they were making the movement, they were on average slower. So there was that correlation in the neural data as well, which is nice because it sort of brought us full circle. We started looking at a correlation in in this original study, which led to a clear hypothesis, we then tested that hypothesis, which kind of confirmed it, and also produced this additional result that we hadn't looked for in the original data. But then when we went back to look for it, it was actually there. Also a modest effect, I should say, because there are all these other complicating factors in these kinds of tasks that affect vigor, like how hard the stimulus was, how sated the animal is it early in the session or later. So if we were to design an experiment to explicitly test the second effect of baseline levels of activity and vigor, it wouldn't be this one. But it's still compelling that it's there everywhere we look for it. And we are thinking of new experiments to corroborate this.
We're doing new experiments.
Yeah.
I guess undersell and overdeliver. That's fine.
I got another question, which is it's not in the main bit of the paper, but in the final supplemental figure. You guys looked at the effect of what happens if you cool the brain for several sessions in a row, and you found that the behavior adapted. So on the first day, you see this big, shifts in temporal, in time-dependent choices. But by the third day, the effect is much, much smaller. So what do you think is going on in the brain? Is the striatum adapting? Is the neurodynamics adapting? Is some of the downstream places that the striatum talks to adapting? Any thoughts?
Yeah, at a neural level, we can't say, but behaviorally, we at least know that they're able to adapt to volatility in the environment and in these kinds of time ranges, because we did this. Once we saw that the effect was going away, we thought, okay, so maybe there's some trivial explanation. There's goo building up around our implants. They're becoming less efficient somehow. All these kinds of not so exciting explanations for why it might go away. But we also considered this more interesting one, which was you're manipulating the animal activity in this brain region, but you're still providing feedback to the animal. So what usually felt as a long stimulus, if you cool down the striatum enough, it now feels like a short one. But you are telling the animal that they're wrong. So one way they might deal with this feedback is to think, okay, so now maybe the environment is volatile, and sometimes the boundary is 1.5 seconds, sometimes it's less than that, sometimes it's more than that, depending on whether you're cooling or warming. And so we designed this task variant where we essentially did just that. We manipulated the boundary. Every 3 minutes, it would change to one of 4 predefined levels to sort of match the design of the temperature experiment. And we saw the mirror image of the temperature effect going away, basically. We saw that early on, they weren't able to pick up on these subtle changes in where the boundary was, but across sessions, they became faster at tracking local feedback, local in time, to better track the boundary, essentially. So you see that psychometric curves in that behavioral manipulation, they're all on top of each other. On the first day, we introduce this manipulation, but then they slowly start diverging as the animals get exposed to this new volatile task.
If I could just add, I think a good intuition for thinking about this is if I gave you a stopwatch, right, and I asked you to do this task, and then I changed the speed of the stopwatch, right, you would start getting some judgments wrong, right? But if I keep telling you you're getting them wrong, then you're just going to adjust your decision making to the new speed of the clock, right? And so, that's what it looks like the rats were doing, and that's why actually perturbations of timing, it's so tricky because you're kind of constantly getting feedback about whether your time estimates are correct or not, right? Just from being. And so that's why it's really important to kind of look in those initial experiments where the animal hasn't had time to kind of learn to adapt, to this way that you've changed their brain circuits. And I think that's something that's general across a lot of different perturbation studies in neuroscience is you don't want to just look at the endpoint after, the animals had a lot of experience with your perturbation, because like I said, brains like a 747, animals are very opportunistic. They will learn, to find some way around your perturbation in many cases, so that was really important here.
I think some of the old literature where they kind of give drugs to mice and rats that cause them to cause this clock, whatever internal clock that might exist, to run faster. They see that if they do it for several days in a row, the animal will adapt. But one test that they can see whether it is consistent with the internal clock being faster is that they would then take it away. So they take the clock speed drug away. So now suddenly the clock is back to slow, but you have learned a new set point. So the animal overshoots the opposite way. So that's one way of potentially getting at, like, did the animal actually learn to adapt to the cooling or the heating if you do it for several days. Anyway, it will be fascinating because often we think of striatum being the learning kind of center of, at least in the basal ganglia field. The striatum is where a lot of the learning is occurring. But if the temperature cooling is changing neurodynamics consistently, but the mouse is adapting, it might suggest some of the downstream regions might be learning to adapt. Anyway, just some thoughts.
So does this mean that people who live in cold climates are responding differently from people in the tropics?
Or just cold-blooded animals?
I just wanted to clarify. Yes, we're not asking how, like, if your brain runs hot or cold, does that change your experience of time? The temperature is used as a tool to affect.
Yes. Sure.
Yeah.
Although so
what are your thoughts?
Yeah. So I mean, there's a famous story about Richard Feynman running up and down the stairs in the dormitories at Princeton, because he wanted to know if he was really hot, would his sense of time change?
Mm-hmm.
But
no way.
This is one of the problems with physicists coming into biology, is that he didn't appreciate that we have lots of systems for maintaining our body temperature at a stable level.
But on a small level.
But there are old reports. There are old reports of people in fever, for example,
right?
Possessing significantly altered temporal judgments. So
and also just perception of reality in general.
But to be clear, like, we are warm-blooded, so no matter what the external environment is, unless you're in hypothermia or fever, your brain temperature is constant. So
sure. No, but fever is a good example. That's you can't respond. You're hot. It would be interesting to see how that affects.
I think fever is actually really interesting. And I mean, there's a couple of sort of more recent papers suggesting that learning. There's even this idea that some autism symptoms go away when you have fever. There's something interesting there, but of course you can't. There, it's hard to divorce the infection, the outcome of the infection, versus the body then saying, okay, this is what we're going to do in response to the infection. Anyway.
Well, you could induce fever in an animal without an infection, and see you could do your experiments.
That's what they did, yeah.
I mean, you did it with a probe, but you could also do it with...
Yeah, it's an interesting idea.
A cytokine or something.
Right, without having to put I don't know. Anyway, That's great. All right, we'll wrap it up now. That's TWiN 53. You can find the show notes at microbe.tv/TWiN. Send your questions and comments to TWiN@microbe.tv. If you like our work, please support us. Microbe.tv/contribute. Our guest today from the Champalimaud Foundation in Lisbon, Portugal, Joseph Paton. Thank you so much for joining us.
Thanks for having us. It was a lot of fun.
And Felipe Rodrigues, thank you, and good luck in your postdoc.
Thank you. Thank you for your interest in our work.
Jason Shepard's at University of Utah. Jason Synaptic on Twitter. Thanks, Jason.
Yeah, thanks to Felipe and Joe. It was super cool stuff.
Tim Chung is at New York University. Thanks, Tim.
Thanks. And yeah, thanks, Philip and Joe. That was very great. I had a great time.
Vivian Morrison is a neuroscience research advisor based in New Orleans. Thanks, Vivian.
Sure thing. Thanks, guys.
I'm Vincent Racaniello. You can find me at microbe.tv. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back next month.